So in the last class, I introduced you to the problem that we are trying to solve with uh, the basic local alignment search tool, or shortened call as BLAST. The idea is you have so many nucleic acid sequences which are continuously being deposited in different databases. And typically you would be starting with a small query. When I say small, it's relatively in relative numbers. So it would be something which is about 1,000 base pairs to 10,000 base pairs. And you want to search this query in a database. And the algorithms that we have used before typically don't give you an optimal solution in the time that you would want to solve the problem in. So we try to take cues from some analysis that we had done earlier, specifically analysis such as these dot plots using dot matcher, where you see that whenever an alignment would be happening between two sequences, you would essentially see uh, a line along the diagonal. So for example, you see that here you see alignment along the diagonal, and typically whenever you would be having an alignment, you would see series of letters common between your query and your database. So query is basically a smaller sequence, and a database is a collection of much more number of sequences, or much larger sequence. So I introduced you to the idea of solving this problem using the concept of indexing. So just like you have a textbook, and the end of the book, you would have an index, and you want to search a sentence in the book, you would essentially try to look for keywords in your sentence which are not very common. So for example, here the name Barrett Joseph is a very uncommon name or uncommon word to basically encounter. On the other hand, climbed a wall, it might, the probability of having this letter is quite a lot. So therefore, typically in an index, you would have locations of these uncommon words rather than the common words like climbed a wall, right? So you essentially look for these uncommon words from the index of your book. You go to the specific page, and then once you have found this word, it is essentially the same idea as finding a series of dots in a dot matrix, and you can essentially extend these dots or this alignment to the left and to the right, and find whether the specific words that follow this unique word actually match the query that you have. All right. So the basic idea for the algorithm is you have to have an index first. So you need to have a pre-processing of your database. The algorithm then is essentially searching the index. And once you have found the index, the location of the keyword that you have, once you have this keyword location in your database on the specific page, let us say, then you would go and extend the letters to the left and to the right, and we will try to match your query sequence with it. So a question that I asked uh, you last time in class, and I think not many of you were confident about the answer. Can you tell me what kind of alignment are we looking at here?
So we talked about like a local
Local
alignment, right?
alignment.
Right? We are trying to look for a local alignment because typically your query is in this case much smaller than your database. So a query is only going to be locally aligned somewhere in the database. Okay. So this idea actually lays the foundation for next set of algorithms, which are quite popular both in bioinformatics and in a lot of computational applications where you need to have these kinds of database searches. Basically, whenever you are dealing with large amount of data, and these approaches are called heuristic approaches. When I say use the word heuristic, the word heuristic essentially means learned from experience. So the idea is that these heuristics are experiences with no clear mathematical formulation. All right. So what is the heuristic in this case? For example, whenever you must be giving your GE, right? You would be looking at the textbooks and they will be also telling you how to solve problems. So sometimes you will be having, in your 4 options, you just plug in all the options into the formula and you basically use trial and error. So you know that whenever you will plug in the value in the formula that you've been given, by trial and error, you will get to the right answer. So you did not try to solve the problem by the knowledge of the uh, solution, but you try to solve the problem by fitting what solution works the best. So that was your heuristic. The other heuristic that most of you would have learned, probably one of the first things that are taught, is the process of elimination, right? So whatever choices look very weird or they are off, you most likely will eliminate those choices. So when we talk about the experience of searching a textbook, right, or aligning a sequence, what is the heuristic that you usually see? Can somebody tell me? So we've already discussed what heuristic it is. I just want somebody to sort of repeat it to me.
So maybe we have like genes are there, so we know that this gene is have this sequence. So if that is matching, then we can say that yeah, this is here, and directly go there and not search like from a start to end, I guess.
Okay. So can you maybe give me a slightly more formal way of saying that?
So I guess like just as we like refer to an index, then we see it's this specific section or this specific page. So rather than searching through the entire book, we can just go to that specific section and we can search through that like
That's the
section
same
itself.
thing. That's the same thing. That's the same thing that Ratnesh is saying. But the basic idea essentially is that alignment, right? The location wherever you will have an alignment, the query and the database is going to share k-mers, right? You're going to have the same k-mers present in your query and your database. Is this clear? So the heuristic that we have
Yes, sir.
is the fact that whenever an alignment is going to be happening between two sequences, that would essentially be based on shared k-mers. So now this is the basic idea of the BLAST algorithm. So let us see how it works. Let us say you have a query given here. So this is much smaller. I mean, it's a dummy example. So this is small query and you have a database. So the query is 6 words long and the database is, I think, about 13 letters long. And we have a scoring scheme. All right. So whenever you see a match, it's a +1. Whenever you see a mismatch or a gap, it's a -1. Same thing that we've been doing. Right. So now, before we actually go and develop the BLAST alignment, let's now first look at what are the real local alignments possible. So we run the local alignment algorithm that we have discussed before and make the matrix. And then when we backtrack, these are the common, these are the alignments that you get. All right. So can you tell me the alignment that you would be getting, say, for the group 3? What letters are going to be common for group 3?
So C
A
A.
and C.
A and C or C or A, right? So now if you were to do this using local alignment, what's going to be the big O notation? How much time are you going to take? So what's the big O for local alignment? How many operations will you be doing to make this matrix? Using local alignment, I'm basically asking you a question from the previous to previous module. It will be the length of the query multiplied by the length of the database, or n squared.
Yes,
Yes,
sir.
sir.
Yeah. Okay. So this is not really good when we are talking about very large databases, right? So what dynamic programming algorithms do, they give you an accurate solution, right? But it takes a long time. What BLAST or what these heuristic algorithms do, they reduce the accuracy slightly, little bit, right? But they are running in much, much faster time. And we'll see how. Okay, so in the case of Smith-Waterman, the accuracy is very high, right? And so is the time. But in the case of BLAST, the time is less, but the accuracy is not as good as dynamic programming. All right. So now how do we actually start to develop this algorithm? So we have the database, which is basically a string of length 13. And the first step would be to create the index. This is called indexing or indexation. So just like you have an index for a textbook, you would want to create an index for the database in which you want to search the words. Now, can you tell me how many times you would need to do this step?
Sir, which step?
The step of indexing.
Sir, whenever we have a new sequence, we will have to do indexing. After that, we don't have to do.
Right? So for a database of a fixed length, the indexing step is going to happen only once. And even subsequently, let us say you have 1 million sequences. If you add a few sequences more, the indexing step is not going to take so much time. Why wouldn't it take so much time? Can you tell me what is the time complexity of indexing? So
The number
indexing,
of days
Will
here
be n, right?
and
yeah. So indexing essentially is creating an index or a dictionary of k-mers found in the database, right? Okay. So the how would you
yes, sir.
extract k-mers? You would extract how many k-mers you can extract, n minus k plus 1. So the time complexity is big O n. So for each sequence you add to the database, it's going to basically be as long as the sequence itself. So that's a relatively very fast process. All right. And for a database for a fixed size, you only have to do this once. So in the case of BLAST, when you'll be looking at working with the tool tomorrow, the length of the k-mer is also called word length because they were borrowing ideas from making a dictionary, like an index for a dictionary. All right. Is this clear?
Yes, sir.
Yes, sir.
All right.
Yes, sir.
So how do we make the index? So you extract k-mers, right? So you go from the left, in this case, you cannot go from any direction, and you start to extract all these k-mers. And now this becomes your database sequence. All right, so you have 11 k-mers that were extracted. N is 13, k is 3 plus 1, right? So 11 sequences. So now you have your index. Now, is this enough? Or do we need to store something else? Is this how an index looks in a book? Or is there some more information in the index?
So we have to align like alphabetical sequence or something like that.
Okay. Besides the alphabetical nature, is there something else that you have in the index besides the word itself?
We have to say
So
that
the
like where this
frequency.
sequence is. Yeah.
Exactly. So you essentially need to tell exactly where this particular words, these k-mers are appearing. All right. So we will do that as well. So now let's go to the second step, which is aligning your index to the query to generate high scoring segment pairs, also called HSSPs. All right. So what do I mean by that? You essentially extract k-mers from your query the same way that you did for your database, right? And now you see which k-mers are appearing more frequently than by chance, all right, in your database. So can you tell me what's an easy way of scoring a HSSP, which is a k-mer which is found in your query and a k-mer which is found in your database? You've already done this.
So could you repeat the question once?
Say that again.
So could you repeat the question once?
So the question is, how will you be scoring the HSSP? Can you come up with a simple way of scoring the HSSP?
Sir,
So
like
based
we
on Hamming
did.
distance.
Hamming distance, right? So Hamming distance is extremely easy. You are given a k-mer, you are given a sequence, and you essentially calculate the minimum Hamming distance, right? Between your
Yes,
query
sir.
and your sequence. And you essentially return the location of where the minimum Hamming distance appears, right? What's the time complexity of this step? Come on, we are towards the end of the course. These are like basic questions that I hope everybody
Sir,
can answer.
n square.
Time complexity is the length of the query, right?
Yes, so sir, n square it should be.
Should be n squared.
It should be n
So it
itself.
should
Should
be
be n, right?
K.
For every number of queries multiplied by the length of the query, right? Of number of k-mers
So it
multiplied by
should
the
be
length.
N into K, right?
Yeah. So n is the length of your query and k is the number of k-mers, right? So how do we do it? We essentially we take each of the k-mers, right? And then we say, or we extract all the k-mers from your query, and then we say which one of these is giving you the best Hamming distance, right? So in this case, the best Hamming distance is with GAT, right? So GAT is giving you a score of 1 because you have +1, +1, -1, right? Given your score and mismatch. And here we define what's called a HSSP threshold. So HSSP threshold is again based on a heuristic. It's the idea that we are not going to pursue, or we are not going to look at common gamers, right, whose score falls below a certain score, right? So the idea is that if it falls below a certain score, most likely it does not represent a local alignment. So if the threshold HSSP threshold is +1, that basically means you should at least have 2 matches in your sequence, right, in between the gamers. Right? This is just a toy example. You can imagine that when you have larger gamers, as you will see in the class tomorrow, these scoring schemes are different. All right. So you take all the gamers and you then run them through the database and you search them through the database and then you do the scoring and you tell exactly which gamers are most are basically above the threshold. So for example, for GSC, it is GAT here. For ACA, right? There are these 3, right? And whenever you're extracting a k-mer from your query, you know exactly where you're extracting the k-mer from, right? So essentially at the specific coordinates, you know that your which of the specific coordinates have HSSPs, right? Which of the specific coordinates have good k-mers in the database. All right. And like that, you make basically a table which gives you the coordinates of the k-mer that was extracted in your query sequence and where they lie in the database, right? Because here this value 4 is essentially given to GAT, right? And because GAT is lying at the 4th position in your database sequence. Is this clear?
Yes,
Yes,
sir.
sir.
Yes, sir.
So the steps are: you first index, you align the query index to the query, right, to generate these HSSPs. You score and you identify the HSSPs and you repeat this entire across the entire query to generate these high scoring segment pairs. All right. So the heuristic principle again, specifically for local alignment, is that a local alignment must contain a high scoring segment pair or a HSSP. All right. So this is the heuristic that we're working with. All right. So the next step is basically you find the locations where these high scoring pairs were existing and you extend. All right. This finding the locations of these HSSPs or the k-merge in a database, which are HSSPs, right? Which form an HSSP with your query sequence. Those are the locations for your alignment. So you seed the alignments, and then after seeding the alignments, you extend on both the directions to the towards the left and towards the right of the sequence. Exactly the same way that we were doing for searching a sentence, right? First we found the seed, exactly where in the textbook that word is found on which page, and then we extended the sentence to the left and to the right and saw whether the next words is what we expect or not. Is this clear?
Yes, it is clear, but
Yes,
like
sir.
when we have sequences like John Barrett or something like, so John Barrett was, we know that this is something which is not common, right? But
Yes.
in DNA sequences, when we will get like a sequence of a fragment, then how will we know that this is not common? So find sequence index for this and not from beginning or from end. So like I'm asking
So
just because
understand.
you are saying
Let me
that
answer. So
go back
basically,
and forward,
yeah.
backward and forward
Yeah.
both.
So what happens is, so think about it. If there are some k-mers, right, which are very unique to a particular organism, right? So we are going beyond what is currently taught. So if it's very unique to organism, what you would want to do is you want to have a separate list of these k-mers in the memory. You want to access these k-mers first. Okay. So this is what these algorithms are also changing all the time. And this particular idea that there are some k-mers which are unique to a certain organism is actually used in another tool, which is essentially used to identify species in a complex mixture of DNA. And I can talk about it in the lab tomorrow. Okay, that tool is called Kraken. All right. For the time being, let us assume that we have all possible k-mers for both your query and your database. Okay.
Yes, sir.
So the fifth step is you seed the alignments, you find exactly where your HSSPs are lying, and then you extend. Okay. So how does the extension work? You start with your threshold score, right? And then you have an extension threshold as well. So for example, our threshold score was 1, right? And you extend, whenever you extend, you'll either find a match or you will find a mismatch, right? So you keep on extending until you go below the threshold score. So we say that we are okay to extend until the score goes below 2. All right. So for example, here you start with 1. You're extending this only if the neighboring letter is also a match. If the neighboring letter was not a match, it was a mismatch or a gap, you will just stop the extension there, right? So in this process, you essentially save a lot of your compute steps. So now let me show you an example of how it's done. So this was our database and this is our query, right? We have this kinds of scoring schemes. Your scoring threshold is +1. Your HSSP threshold was also +1, if you remember from the previous steps. All right. So now let us do a seed and extension alignment for specifically this HSSP. So you have an HSSP ACG. It is found at the second location of your query and it is found at the first location of your database. All right. So how do we do it? We extend because we have to find it in the database. We extend to the left side. When we extend to the left side, because it's a gap, so the score becomes 1 minus 1. It is below the threshold. So we abort the extension. Okay. The next, we
Yes,
go
sir.
to the right side. We find a match. We increase. Then we extend further. There is a mismatch. We decrease the score. And as soon as we decrease further, we extend further. The score goes beyond below the threshold and we have obtained our alignment. Okay. Is this clear? I'm stopping here because I want
Yes, sir.
everybody to be absolutely clear about the blast alignment. This is one of the basic ideas of that is used a lot, right? So I hear only Ratnesh's voice. Everybody else is clear?
Yes, sir.
Could you just go over this part once more?
So we start, let us say this was the table that we had, right? Where we have the HSSP pair. So we know that this particular k-mer is found at one location in the query. So this particular k-mer in the query is found at the second location, and it is found at the first location in the database. So this was our fifth step, right? So we have now start a seeded alignment. So now we have seeded our alignment like this. We know that the second location of the query is going to be aligning with the first location of the database. Now what we do is we extend. We first start to extend to the left side of the query, right? We extend to the left side of the query and we extend to the left side of the database because we were at the first location. So there is no letter here. So it essentially means you have inserted a gap here, right? So initially your score was already, so you have a third thing here, a third column, which actually it gives you the HSSP score. So you already started with a score of 1. Now, because you have inserted a gap because of extension, you subtract 1 based on your scoring scheme and your score falls below the scoring threshold, which is +1 here. Okay.
Yes, sir.
You do the same thing now to the other side. When you extend, you find a match. So you have +1. You further extend, there is a mismatch. So you have -1. And then you extend further. There is again a mismatch because there's a gap now, which because of the gap now the alignment falls below the threshold and you stop. You do the same thing for all the HSSP pairs. Okay. So what does this mean essentially? You had the seed, right, at the second location. You have second location of the query and first location of the database. What you did initially was to extend in this direction, right, of the database. And after that, what you were doing was you were extending at the other directions of the database. Okay. So now when we actually compare this with your original alignment, the local Smith-Waterman alignment that we had, you actually get very close to the real alignment. Is this clear?
Yes, sir.
Yes, sir.
So you do the same thing across all the HSSP pairs, right? So therefore, in order to do this, what does the blast need? Blast needs a word size. So when you're tomorrow in the class you will be doing, you will be seeing that in the default parameters, there would be a word size for blast, which essentially means that what are the sizes of seeds that you're looking for to start your local alignment. Then you will have something called the HSSP threshold, which tells you which k-mers or which words to start the alignment from. And then you'll have a second threshold called the extension threshold, which essentially tells you that you should stop the extension once the score falls below the extension threshold. All right, so these are the three parameters that the BLAST algorithm expects from you. So if you have a database of length n and you have a query which is of length 3m, right? The seed length is, let's say, m. Your T1 is approximately, not approximately, it's m by 2. And your T2, its extension threshold is m by 4. Match is +1. Gap is -1, gap mismatch. What is the maximum expected size of the local alignment matrix? This is something that I want you to go and try to solve. And we can discuss this in the class tomorrow or on Thursday. Okay, I hope this problem is clear to you.
Yes, sir.
Yes,
So
sir.
your
Problem
database
is
is
clear.
of length n, the query is length 3m, your seed length is m, the two thresholds are m by 2, m by 4, match is +1 and mismatch is -1. So what is the maximum expected size of the local alignment matrix? So you are essentially seeing that whenever you are doing the alignment, you're not required to do the entire alignment here, right? So you have reduced the computation from doing it only for doing it for the entire database to only a specific region of the database.
So sir, like local alignment matrix will be
Much smaller.
wherever it is matching.
Yes, only
Yes.
wherever it is doing this extension kind of a thing. So
Okay.
it will
Okay.
definitely, the local alignment matrix would be of a minimum size would be a specific size, and the maximum size would be another specific size depending upon these parameters that are user defined. So that's what I want you to see. What is the minimum size and what is the maximum size that you can expect of this local alignment matrix? Okay. So now,
Yes, sir.
once you have this, how is it actually implemented? What are the basic ideas behind the implementation, and maybe we can also speed the process even faster. So that's what I'm going to discuss about next. But before that, I hope the basic idea of BLAST is clear to everybody.
Yes, sir. Basic idea is clear.
Yes, sir.
Everybody?
Yes, sir.
Sukhchand, yeah. Dev, Deepak.
Yes, sir, it is clear.
All right. So now what you essentially have for the algorithm is the input is a query sequence, and you have a target database which has chromosomes, other bacterial sequences, viral sequences, right? You have a huge database. And then you have these parameters. So one set of parameters would be a scoring matrix essentially. So this scoring matrix is again derived from either Blossom, PAM, whatever you can choose this, right? Just the same thing that you had with your pairwise alignments and multiple sequence alignments, right? You use the matrix according to what kind of query or what kind of database hits you are expecting. So for example, if you're expecting to find database hits which are very close to the query, so you would choose what kind of Blossom matrix. You will choose a Blossom, say Blossom 80, or you'll choose a Blossom 45 or a Blossom 62. If you want to find query sequences, you want to find the database hits which are very close to the query sequence.
So then Blossom 80, right?
Everybody agrees to this? So think about it. We'll discuss this in the class tomorrow when we are doing the practical. So this is what we have. So the first steps are pre-process the database, right? Next is find HSSPs between the k-mers of your query sequence and pre-processed k-mers in the database. Next step is find all matches, locations between the query k-mers and the database k-mers. Seed and extend matches. Evaluate the significance of matches, right? So this is the one more step that we have added, which is basically telling us basically we have to find out whether the match or the score that we have got, is this significant or not? Is it better than what you would expect randomly by chance? So this is one more parameter that BLAST tells you in the output. All right. So let's try to implement it. So first process is pre-processing of a sequence database for BLAST. And here what we do is we try to come up with an efficient way of storing k-mers. So the efficient way of storing k-mers is to actually create a numerical representation of k-mers in uh, in a base 4 configuration. So essentially what I mean by that, so if at a specific location you have a, you give that location value 0. If it's C, you give it value 1. G, you give it value 2. If you have C, you give it value 3. Then you multiply the positional value of the specific k-mer by by that specific position value and the value of the letter that is found there. So for example, if you have a k-mer, a former TAGC, so the least significant location will be having a value of 4 to the power 0. Second one is going to have 4 to the power 1. Third one 4 to the power 2. fourth one, 4 to the power 3, right? So if you have a Kmer, the most significant position is going to have is that value is going to be multiplied. That value, the new value of that nucleotide at that position is going to be multiplied by 4 to the power k minus 1. So when you do this, you essentially transform the Kmer into a numerical value. All right. So in this case, this numerical value is 255. This is called a key. Now, are you getting some flashback of what we had discussed earlier? Anybody remembers the hash table that we had talked about initially?
Yes, sir.
So essentially what we are trying to do is we are trying to create a hash table for our Kmers. All right. By giving
Sir,
each.
one question, sir. Sir, why we are counting from right to left and yes,
We
why
can do it.
we are counting?
We can do it from left to right also. It's just a convention.
Okay, okay.
Yeah. Okay. So we can process the database in such a way that the Kmers can be searched with a unique key, as you would do in the case of a hash table. All right. So how does it look like? So in this case, the word length is 3, right? This is what we have specified. This is our query sequence. Let us say this is our database sequence. We take all the Kmers extracted from our database. All right. So these are the Kmers that are extracted from the database. So now we are actually finding that some Kmers, as the database becomes longer, some Kmers are appearing more than once. Earlier, you had one Kmer appearing only once, right? Now what we will do is we will convert all these Kmers into their corresponding key values. All right. So I hope everybody can convert a Kmer into a key value given a scheme. Yes.
Yes, sir. Yes.
Yes, sir.
So the next thing what you do is
Yes, sir.
you would sort the keys in alphabetical order, or in other words, you can just sort them by key value because A is having the lowest, right? And T is having the highest value because of this reason, right? So now this becomes in your lexicographic order, right? You can imagine these same ideas are even implemented when you are doing things like De Bruyne graphs, right? Where a lot of these things would be lexicographically available. Now you have these different key values, you have these different positions. Now what you do is you do something interesting. Either you can have this when you are searching this matrix like this, the search time is going to be the same as the length of the hash table, right? So this is your hash table. We can do some interesting ways of representing this to speed up the process of search, and I'll show you how. So what you can do is you can you can use a data structure which is often used in computation, in computer science, is the data structure is called a binary tree. So how does that work? You start with the middle portion of your hash table. So the middle portion of the hash table has a key value of 26, and this particular Kmer is found at the 4th location in the database. Now what you do is you populate the left and the right side of your tree. How do you populate it? The left side of the tree would be representing these values from 6 to 20. The right side of the tree will be representing these values from 33 to 61. So what you do is you further separate the left side by a recursive algorithm where you go and you say, okay, this part, the first part, I'm going to make a tree for the first part as well. How will I make the tree for the first part? Just like I started making the tree for the entire table by going and adding the middle value. Okay, so the middle value here is 15 for this part of the tree, and the middle value is 40 for this part of the tree. Right? Now you do this recursively. You go down to the left part of this key and right part of this key, and you populate the other leaves. Right? And this is the tree that you get. Now, why did I do this? when you store your hash table in this kind of a format, the searching becomes extremely fast. And I'll show you how. So let us say you have your query sequence, you have your database sequence. You are going to extract the k-mers from your query sequence. And now what are you going to do? You have already pre-processed your k-mer from the database. So you have to now go and search for these query k-mers in your database k-mers, right?
Yes, sir.
Yes. So now we are not going to search it in a linear fashion, but we are going to search it in our binary tree. Okay.
Yes, sir.
So these are the,
Sir.
yeah.
Sir, but like if binary trees are there like this, and how many rows are here? If the rows number of rows are like 4 into 4 into 4, 64, then uh, there is no point of searching. Like we cannot find it. Like I
I'll show
say.
you. I'll show you. So this is how you will be doing it if you don't have a tree. And I'll show you how would you be doing it when you have a tree. Okay. So let us say these are the k-mers, right? These are the keys that you have. So you have to search for the key of 13. All right. How would you search the key 13? You will go to the tree. You will say 13. 13 must be lying on the left side of this tree, right? Because it is less than 26. So you
Yes, sir.
go here. 15. 13 must be lying on the left side of 15 because it is less than 15. You
Yes,
go to
sir.
12. 13 was actually has to be at the,
There is no.
it should be at the right side of 12, right? Because it is greater than 12. So you go here
Yes.
and it's not present anymore. So instead of going through the entire database one by one and matching for 13, you now traverse this tree and only within 3 comparisons you have found out whether the key is present or not. Same thing we will do with 54. 54 is going to be lying to the right side of the binary tree. It's going to be right side of 40. It's going to be left side of 58, right? And it's going to be, it's not found. All right. So essentially what you will be doing is you will be forming all possible SSSPs in this fashion. So you're going to go down the tree and you're going to essentially be forming the doing the scoring for each of the k-mers that you're obtained. Now, once you have done this, you're going to seed the alignments just like you did before. So this is one seed. This is the second seed, third seed, fourth seed, fifth seed, right? And now you're going to extend like we did before. And then we're going to basically come up with the alignments. So what is the time complexity? So you essentially have one operation of making the tree itself. So there are 4 to the power k different kinds of k-mers that you have. You are going to be from your database of length L, you're going to extract L minus k plus 1 k-mers, and not from the database, from your query. And from the database itself, you are going to have n minus k plus 1 different k-mers. You're going to have that organized in a tree, which is a binary tree, and searching a binary tree takes log base 2 multiplied by n, right? So basically the time complexity for BLAST now becomes when you are storing your um, database in this kind of a tree structure, it goes from searching from n multiplied by L, right? The query sequence, it goes to uh, L, your query sequence, multiplied by log of n. So it's an extremely fast operation. So how do we do it? We basically create a database, which is essentially a good data structure for storing k-mers. And we have an efficient algorithm for using these k-mers. So this gives you basically this is the algorithm for BLAST. It allows you to search a query of length n in in a database of length m in n multiplied by log m time, which is quite efficient, right? And the extensions are based on some pre-established parameters, and these are basically locally they will be n squared, right? Length of the query. So the next question is, in this particular case, you obtain lots of matches, right? Some of them look nice, right? Such as these two matches, and some of them are not that nice. For example, this alignment or this alignment, right? So how do we say which alignment is better than the other alignment? Right? So this is what I mean by the fourth step, which is figuring out the significance of the matches that we have obtained. Is the alignment that we have got is a good alignment or not? All right. So before going to that part, I hope whatever I've talked about till now is clear. The basic idea of how BLAST
Yes, sir.
works
Yes.
and how the algorithm is actually implemented for BLAST, the original one.
Yes, it's
Yes.
clear.
So now imagine to answer Ratnesh's your question, if there are certain k-mers which are now sort of represented k-mers, right, for particular species. So you can actually have two kinds of trees. One tree could be the representative species tree, and the other tree could be your k-mer tree, right? Generic k-mer tree. So if you want to ask the question, just tell me from which species this particular sequence belongs to. You might want to just search for the species specific k-mer tree. All right, because you might find k-mers which are unique to their query and to the specific species, and that could be even faster. But if you are having a generic question, right, give me the alignment, then you might want to search for the entire, like the tree with all the k-mers. Okay, so now when we are trying to talk about significance, we have to think about certain questions. So the first question is, are we finding just random alignments? So what do I mean by random alignment? Can somebody tell me? What would be a random alignment?
Alignment which doesn't have like biological significance. Like if we have a gene, then it should be matching with that, and yeah, and
So
like
can you do it without biology? Can you give me what would you, how would, so if I have a database of 1 million nucleotides, right? And if I give you a query of length 4, would you find an alignment for this query of length 4?
yes, sir, definitely.
So what is the, so the, would this alignment be just random in this case? What is the random? How is the randomness driven by? What is the randomness driven by here? So the probability of finding your query is inversely proportional to the length of the query. Can I say that?
Yes, sir.
Everybody agrees? If a query is short just by chance in your database, you might just find some random combination of nucleotides which looks like your query,
Yes, sir.
Yes, sir. Yes, sir. Yes.
right? So this is what I said. If you have a longer query sequence, you would expect to find it more often or less often in the database. I asked the other question now.
Is it less often?
Less often, right? So here by random alignments, I want to basically say that if whatever alignment that you're seeing, is that actually something that you would not find by chance. All right. So this essentially, this idea is represented in a scoring value called expectation value. I'm going to talk about it in the next slide. If the database is longer, does your expectation to see a query increase or decrease?
Decrease.
Decreases, right? do you expect to find a low scoring alignment more easily than a high scoring alignment?
Yes, sir.
Yes.
Right? A low scoring alignment would essentially mean that the alignment is not that good.
Yes, sir. Yes, sir.
Right? That essentially means that it's all over the place. And then what else would you expect to affect the chance of your alignment besides the query length and the score of the alignment?
Also, the distribution of that kind of alignment, if it is available in that sequence or not.
That you do not know beforehand, right?
Yes.
So
database
here
sequence
the expectation value or the E value, it's called the expectation or the number of times you might randomly see this alignment with a specific score is given by M, which is the length of the database. So if the database length changes, the expectation value will change. The length of the query N is directly proportional. So if the length of the query is more, then the expectation value is going to be more. Then there are two constants K and lambda, which are basically dependent on how you have created your index, basically of your database, and S is your score. All right. So E values are essentially you will be giving E values something like this. So if a E value is less than 10 to the power minus 100, so that means there is 1 in 10 to the power 100. Not this is not minus 100. This is 1 into 10 to the power minus 100 or 10 to the power 100 chances, which is almost zero chance of seeing the specific alignment with the given score just by chance. So this means that the specific sequence that you have found in the database is really unique to that organism where you have found that sequence. If the e-value is between 10 to the power minus 50 to 10 to the power minus 100, it essentially means that the expectation is 1 in 10 to the power 50 sequences that you might find an alignment. So this typically represents alignments which are between nearly identical sequences. And this is typically considered to be a good result, and you must scrutinize this further. If the e value is 10 to the power minus 5 to 10 to the power minus 50, that means 1 in 10,000 sequences, you might actually find an alignment like this, right? 1 in 10,000 sequences in the database, you might just find alignment. So this alignment typically represents homologous sequences of proteins from related species. So this is a good result again, and it must be further scrutinized with more exact local alignment tools. So typically you will get a result. Okay, this particular your query sequence was particularly found with a certain expectation value, say 10 to the power minus 10 in a specific species. So then what you will do is you will take the specific sequence where it was found and your query sequence, and you will then run one of your local alignment tools to know exactly the score and the alignment values. If the expectation value is 1 by 10 to 1 by 10,000, then in that case, these alignments are um, like as very likely you would say, and they might represent very distantly homologous sequences. So maybe you are trying to your query sequence might be from an insect, and you are trying to find the same kind of sequence, let's say in a bacteria, which are millions of years apart. And if the e value is less than zero point 0.1 is greater than 0.1, that means there is a 1 in 10 chance that the alignment is going to be found in your database, and it's most likely a very short fragment, and it is a random alignment. So this is not a significant value. So when you actually go to BLAST tomorrow, when you'll be doing it, you'll be seeing that BLAST is essentially a collection of tools. So there is different kinds of databases that are behind these BLAST tools. So the most popular one is called BLAST N, where the database is a database of nucleotide sequences. Then there is something called BLAST X, uh, which is basically you translate the nucleotide in all possible 6 frames. So the nucleotide database in all possible 6 frames to get amino acid sequences, and you obtain a database of potential proteins. Then there is T-BLAST X. So in this case, the query is translated in all 6 frames, and the database is also translated in all 6 frames, and these two things are matched. And then there's also the simple version of BLAST P, which is the queries in protein sequence. So this is an amino acid sequences, and your database is also in the amino acid sequence. All right, there are many different versions of it. And in the class tomorrow, we'll be exploring all these different versions, and I'll be talking.
Sir, can you please talk about what is this 6 frames?
So given a nucleotide sequence, how many different kinds of proteins can it, how will you find out what proteins can it make?
Based on the genetic code?
Based on the genetic code, right?
Yes.
So if you don't know the start codon, all right, what is the start codon?
ATG or AUG.
Right?
Yes.
So if you don't know where the start codon is located or where the protein translation is happening, so if I give you a DNA sequence, how will you find out what amino acids it encodes for? How many different ways in which can you translate a DNA sequence that is given to you?
Okay, sir. Like understood. Like we have frame shift kind of thing. So that's why it is 6, I guess.